Hi everyone, I'm Montse Murel, and I'm the Director of Applied Research and Innovation here at Syntego. In this second session, we will focus on how CRISPR-based cell mothers are really enabling researchers to advance their understanding of the most challenging to study diseases. We will hear from leading experts of how CRISPR is accelerating and improving disease research. After this very interesting session, I want to remind you that we'll have more entertainment lineup that you don't really want to miss. It's a pleasure now to introduce the chair of this session, Dr. Laura Lambert, who is an assistant professor and director of the Transgenic and Genetically Engineered Models, TGEM, core facility at the University of Alabama, Birmingham. She uses state-of-the-art methods to manipulate the genomes of rather modern systems. She has extensive experience in the replication of genetic conditions and modeling of patient-specific mutations related in diseases so important as breast cancer and cystic fibrosis. As well, she characterized the pathogenesis of these genetic disorders in rodents. These models, of course, are essential to research for the discovery of novel therapeutics and understanding the role of genetic variants. Dr. Lambert, thank you so much for accepting our invitation to be the chair of this session today.
Hey, thanks so much for having me as the chair and inviting me to give a talk today. So our first speaker, it's my pleasure to introduce to start off this session, is Emanuela Cuomo. She leads the Target Validation UK team in early oncology and is part of the selection team and DepMap collaboration. Emanuela is interested in the application of innovative methodologies in precise genome editing to study gene function and disease and mechanisms of resistance to targeted therapies. Emanuela?
Hi. Thanks for the invitation. What I'm going to do today is really focus on target discovery in Astrazeneca. We're using genome editing actually quite heavily from developing new tools, new methodology to use for developing preclinical models, understanding targets, discover and identify new target by CRISPR screens. So we're really heavily involved in genome editing. But what I want to focus today is really on how we use genome editing for target validation. And recently we have been really changing the way in which we identify and suggest target across disease area, but today I will focus on oncology. Previously, the way in which we tended to, the sources of new targets were really from collaboration, from literature, mining existing and public data set. But today we are seeing new targets coming from functional genomics, both internally and externally, from AI and machine learning, and also from data coming from patient and medicine and targeted drug in the clinic. Now, those targets are not necessarily well known, and there is sometimes not a wealth of information on them. And so we really need to validate those targets before we enter them in the portfolio. One of the main reasons is also that one of the main reasons for failure of clinical trials is actually a lack of efficacy. And that is very much based on the fact that target may not be very well validated. In these slides, it's really just to illustrate the failure that we often see in clinical trials with some of our drugs, and the reason why we really need to better validate our targets. Right. So this is really just, I'll skip quickly. This is just to, this slide's okay, thank you. So this is just to focus on the target validation pipeline in our drug development. And the example I will give today, can you go a slide back, please? the example I will give today, it's really focusing on how we try to establish the link between a gene and a disease, how we use genome editing to validate this link, and how we actually also use genome editing to understand resistance to target therapy, so how gene loss, gene variants can affect the response to target therapies. And the last example will be about how we try to predict what type of response we have in the clinic with our drug. So if you stay on this slide, here is actually an example of devalidation. So a specific gene supposed to be essential for small cell lung cancer, for non-small cell lung cancer, line with a KRAS and LKB1 mutation. And so how you can see here that we used cell lines, lung cell line with different genetic background to knock out this gene with two guides. And you can see at the protein level and the messenger level, we see reduction of expression of this gene. But on the bottom left, you can see that actually compared to control, we don't have any effect on the viability. So although this dependency was published using RNAi, by CRISPR knockout, we show that this gene is actually not essential in this genetic background, and therefore we didn't bring it in our portfolio as a new target. If you can go to next slide. And so this is really the basic type of work we do, but then we do also more specialized type of work. Can you go to next slide? Yeah, thanks. In which we try to understand actually patient data coming from resistance to our target therapies. And here you can see an example of a tyrosine kinase inhibitor, Tagrisso, that is used in lung cancer patients that have EGFR activating mutations. Can you go to next slide, please? And I have previously presented this data, but I would like to start from here to just illustrate what we do in those cases. We have a number of inhibitors for EGFR, but unfortunately, a patient can develop resistance, and in some cases, this resistance is actually due to a mutation in EGFR. And so novel inhibitors have been developed. Can you go to next slide, please? Novel inhibitors have been developed for these, and one of the things that we are trying to demonstrate is if, for example, those on-target mutations, and some of those you see illustrated there with a small molecule inhibitor that is an ATP competitor, if some of those mutations that we find from sequencing patient biopsies are really causing the resistance. Can you go to next slide, please? Next. And in the next slide here, I illustrate how we input activating mutation in cell line and then specific variant coming from patient data, and we show that this actually results in specific resistance to specific inhibitors. Can you go to next slide, please? We have also tried to accelerate this work by really trying to understand and predict other mutations. And here I'm illustrating a case in which we look at specific amino acid and we input every possible variance, every other amino acid. And like this, we show that we can actually predict clinical data, and we can understand which specific variant will actually cause resistance. Next slide, please. And we have brought this work even further, analyzing a number of clinical data like shown here, additional mutation causing resistance to tyrosine kinase inhibitor. Next slides, please. But also trying to do this type of work really across targets and across genes by mutating every single amino acid and creating every single possible variant and asking the question if these cause resistance to a specific drug. Next slide, please. And I just have a quick example to show, and these are proliferation curve. When, you know, in an assay like this, in each well, you have a specific amino acid and every possible variant for that amino acid in EGFR. And you can see that clearly, for example, in exon 20, we're able to identify only the mutation and the variants that have been actually found in the clinic. If you go to next slides, please. And so this allows us to really look across a gene. But also in the patient population, we found variants and gene loss across many genes. And so another setting in which we use genome editing is actually to test this loss of function by gene knockout in parallel, in which we look a long-term assay like viability upon drug treatment, or even short-term looking at downstream signaling or apoptosis assay. If you can show me next slide, please. And here is again on resistance in EGFR-driven tumors resistance to osimertinib. And in this next slide, you can now see that we have analyzed a number of genes, and some of these have really confirmed to confer resistance. And the difference between doing these and a genome-wide CRISPR knockout is that we're really validating here data coming from the clinic. If you can go to next slide, please. But the next and last bit that I really want to show you today is that we are also not only trying to look at variants in the resistance setting, but we have also tried to understand and to use genome editing to understand protein domains, their relevance in the signaling of the protein and their relevance in cancer. If you can go to next slide, please. And here I'm focusing on the RAS pathway, and specifically on the RAF genes that are central to these pathway signaling. They form homodimer and heterodimer, and they signaling from KRAS to downstream. Now, those genes, specifically BRAF, are commonly mutated in cancer. And on the right, you can see that across BRAF gene, you have a number of mutations that are arising in different type of tumors, and these are across both phosphorylation site that are important for the regulation of this protein, but also the kinase domain and other conserved domain. Now, the relevance of some of those mutations, the functionality has been studied, but not all. And some of those domains have been studied in their importance for dimerization, both homero and heterodimerization, but not everything is known. And so we have used genome editing. If you can go to next slide, please. We have used genome editing to try to look at the functionality of those domain and mutation. And we have designed basically base editing guide across both BRAF and CRAF genes, as you can see there on the right. And these cover quite well the known domain, but also some less known regions. And the results I'm showing today are really focusing on a subset of those guides, because this is the first time we do this type of work. And therefore, we really focus on a subset of guides. And this was done in collaboration with Synthego, as they provided those guides for us. Next slide, please. And this, I think, it's really my final slide, in which I want just to kind of, from the method point of view, highlight that with base editing, we find, depending on the cell line, very high efficiency of being able to input mutation in a gene. But what we also find is that in the editing window, if there are more than one residue that can be mutated, we often find that. And also that we can really, due to the high efficiency of genome editing, really scan a gene and assay different downstream property. And for this, we have looked at homo and heterodomains, and we have also looked at downstream signaling, as I'm indicating here for CRAF. And we can look at the amount of protein by immunofluorescence. We can look at downstream signaling by immunofluorescence, and we confirm some of the known relevance of some amino acid in downstream signaling, but also we find a novel relevance that we are gonna study further, and by comparing downstream signaling with the total amount of protein, we can also infer which are the amino acid that variants that actually result in less stable protein. If you can go to next slide, this is really the end of my talk, and I just want to briefly acknowledge all the colleagues in Astrazeneca, but also all the collaborators that have made a lot of this work possible. Thanks. I'm happy to reply to questions.
Thanks, Emanuela. I think we'll take one question. There's plenty in the chat if you have time to get to them later. Arun Bat wants to know, how do you control expression levels for different mutants so that it's an apples to apples comparison?
Okay, so we tend to do that by always checking both messenger RNA and protein level, we have noticed that genome editing can result in differences, modulation of protein, sometimes, especially with CRISPR-Cas, due to the fact that you may not only input a variant, but actually also inducing a knockout or an indels. So we tend to do that by messenger RNA and by protein levels, Western or immunofluorescence, as in the last case that I have shown. And we only draw conclusion by actually normalizing for protein levels.
Okay, great. Thank you so much. There are other questions in the chat, and then we'll get started moving into the next talk. So the next talk is our first joint talk by Nevin Krogen and Jacqueline Fabius. Nevin Krogen is a molecular biologist at UC San Francisco and director of the Intensely Interdisciplinary Quantitative Biosciences Institute in the UCSF School of Pharmacy. And he is a senior investigator at the Gladstone Institutes. And Jacqueline Fabius is the chief operating officer for the Quantitative Biosciences Institute, where she heads a number of initiatives, including establishing relationships and collaborations, as well as media and communication strategies for the Institute.
Great to have an opportunity to tell you about some ongoing work here at the Quantitative Biosciences Institute. And we're going to try to highlight collaboration and in often cases how we're using CRISPR to facilitate that collaboration. So what I'd say here on the left is really the scientific vision of the Institute, and really my lab, is trying to put together different technologies in an integrative way and focus those technologies on a variety of different disease areas to get at the underlying biology across multiple disease areas to ultimately point in therapeutic direction. So we're heavily involved in combining mass spectrometry or CRISPR-based genetics, structural characterization, mostly cryo-EM and chemical biology approaches woven with bioinformatics. And this pipeline is being focused at a number of disease areas in the context of these different mapping initiatives. So the cancer cell map initiative, the psychiatric cell map initiative there on the right, and then the host pathogen mapping initiative, And we focused a lot on a variety of different pathogens. There I see Jacqueline is back. And we generate these protein-protein interaction maps, including HIV, which I'm going to speak about in just a minute, hepatitis B and C, different flaviviruses like dengue and Zika, and SARS-CoV-2, which I'll tell you about in a few slides. I'm going to let Jacqueline tell you a little bit more about the Quantitative Biosciences Institute first.
Yes, so we thought we'd give you a bit of a context of the Quantitative Biosciences Institute and how we've really set up a platform for collaboration and the various stories that we're going to tell you about. So the Quantitative Biosciences Institute, QBI, is comprised of 129 labs. It's collaborative research, which is quantitative, it's discovery science. My slides have gone to animal models. I'm happy to say I think we might dive into this a little bit later in our slide deck, but perhaps if we could go back to the QBI. Thank you. So, the research is collaborative. It's quantitative, discovery science, disease agnostic, experimental, and computational. As Nevin mentioned, we're very well known for our cell mapping initiatives, which you see up here on the right. And however, we're very spoken about for investing in the young in science, and whether that comes in the form of creating, for example, the QBI Scholarship for Women from Developing Nations, or specific fellowships that take people from the beginning of their career into a platform, which we'll speak about shortly. However, we're most known for our events that are quite creative, and our international and joint symposia, which have been sort of the cornerstone of many of our collaborations around the world. And the QBI Fellowship, which I recently mentioned, is going to play a role later in what Nevin's going to talk about in the context of the QCRG, the QBI Coronavirus Research Group. The fellowship identifies promising standout candidates and speeds them up from graduate student to independent PI. And it's really a 2-year fellowship with an office space, a small lab space, and funding to be creative with. And this particular person actually has become quite a star in our COVID research at this time, and Nevin will talk a bit more about his research. We also started the QBI Scholarship for Women from Developing Nations in Biosciences. And really, this is an opportunity not only for capacity building, but for really planting someone somewhere else to start collaborations across borders. And in this case, Jackie Lugemwa is from Uganda, and she's currently working with us on COVID as well as other initiatives of ours. The collaboration culture at home started the ground up, where we started with something called the QBI Happy Hour, where each month 3 labs nominate 1 postdoc that have 5 minutes and a maximum of 3 slides to share their work and start talking with beverages and hot food. And we found that there was a tremendous amount of collaborations that started within the campus in these 129 labs at that point. So as we mentioned before, we have a scientific vision of collaboration through the cell maps, but and Nevin talked about these maps, which he's going to dive into quite shortly. And we started thinking at the same time, well, what if? What if in the same way we map cells, we started mapping the world with collaborative interactions? What if cell mapping equaled world mapping? And what if protein-protein interactions, in fact, also equal people-people interactions? And what we found as we started mapping the world, we started looking at where we were going and what interactions we were creating with different people, institutes, academic or industry. And we started finding that, in fact, people started falling into very specific networks, much like the protein networks that we've been talking about. And people grouped around whether it was cancer or technology, infectious diseases, and such. And importantly, for our upcoming story on the coronavirus research group, you'll notice that both New York and Paris were already in our infectious disease network. And at this time, we have a number of collaborations around the world and still growing. We sort of feel like the world is our oyster, I suppose. And so we have a number of places that we've been interacting with, ranging from Haiti to Uganda to China, and really very fruitful collaborations, especially in times such as the pandemic. And so the formation of the QCRG, the QBI Coronavirus Research Group, was really the understanding of the context of a coming pandemic and taking action. However, really, when we look back, how did it all start? It actually started with HIV research, which led the path for the SARS-CoV-2 research. And Nevin's going to tell you a little bit about this.
Right. So as Jacqueline said, the foundation was really laid. And I think that's an important point to make, is all the research money that went into studying HIV and other viruses was so important as many virologists pivoted to study SARS-CoV-2. So as we said, we're in the business of generating protein-protein interaction maps. On the top left, the first map we generated with the pathogen was HIV. So we generated an HIV human protein-protein interaction map. we identified 435 human proteins that physically connected to at least one of the HIV proteins. And we wanted to do was to try to functionally validate those interactors. Did they have any effect on HIV infection? But Judd Hultquist, a postdoc in the lab who is now a professor at Northwestern, collaborated with the Alex Marson lab here at UCSF, and he set up a system where we could use CRISPR to knock out human genes one at a time here in primary T cells, the most relevant system for HIV. And here's the pipeline that he set up, and then we could knock these genes out and then infect the cells with HIV and see what effects the knockouts have. And what we wanted to do here was essentially not just knock out one gene, but knock out all 435 genes whose proteins we found physically connected to at least one HIV protein. So the goal here was to try to set up a high throughput 96 well format a pipeline in which we could do this. And Judd and Joe Hyatt, a student that works jointly with myself and Alex Marson, did just this, where we're taking multiple donors using a CRISPR-Cas9 RNP system, getting that into cells, knocking out genes, infecting with HIV, GFP HIV, so we could look at monitor HIV infectivity. Did this at days 3, 5, and 7. And then importantly, we're also sequencing all of the cells to see how effective the CRISPR knockouts were, because we wanted to correlate infectivity with CRISPR knockout efficiency. So here's a picture of Joe late, it was like 3 in the morning one time, with the picture that he took, that I think Judd took of him. And this just represents a relatively small slice of the reagents that went into this particular study. So as I said, we were sequencing all the cells that we tried to knock out so we could generate these type of graphs where each node corresponds to a gene. On the x-axis is editing efficiency as determined by sequencing. On the y-axis is the effect on HIV infectivity. So what we're interested in are the things in the top right-hand quadrant, genes that we know we could knock out that have an effect on HIV infection. So if we go back and look at our protein-protein interaction map, 435 human proteins, like I said, we tried to knock them all out. We got most of them knocked out. And 86 of them, when knocked out, had a statistically significant impact on HIV infection. Since it was quantitative, we could find dependency factors and restriction factors, genes that when you knocked out, infection got better. So in blue here are genes that are dependency factors. In red are restriction factors. And overall, we got about a 30% validation rate in this particular cell system, which is incredibly high. As a comparison, what we did was just an RNA-seq experiment, as many do, in the same cell system. When you infect with HIV, we took the top 75 genes, upregulated the bottom 75 genes, downregulated, knocked them all out, and we got 1 out of 150 that had an effect. And in fact, it was the same one that was on our protein-protein interaction map. To me, this illustrates the power of looking at protein-protein interactions to find biology. So using this type of logic, we pivoted quickly when SARS-CoV-2 hit in the context of this QCRG that Jacqueline alluded to, the QBI Coronavirus Research Group, involving many different groups at UCSF, and involves many different academic institutions around the world and a number of different very exciting industry partnerships as well that we've been working with over the last year and a half. So we were the first group to clone out each one of these genes, the SARS-CoV-2 genes, about 30 of them. So what we wanted to do is, like we did with HIV, put affinity tags on them, purifying them from cells, and then look via mass spec, what are the co-purifying human proteins? And this is one of the first maps that we generated carrying out this particular process. The red nodes are the viral proteins, the circles are the human proteins. So in total, we had 332, a list that we gave to Brian Shortcat and Kevin Showcat. And then they gave us a list of compounds and drugs that would bind to and inhibit these set of proteins, 69 in total, that we would hope would be enriched for the virus to be needed to infect our cells. So we collaborated with New York and Paris, as Jacqueline told you about, and found that many of these did have antiviral effects. Based on all our work, there's actually 26 drugs in clinical trial right as we speak being tested for COVID-19, 2 that we're really intimately involved in, 2 translational inhibitors, Aplidin and Zotadefin. Aplidin, which is also known as plitidepsin, is from the Spanish company Pharmamar. It's actually in a phase 3 clinical trial right now at 70 sites in 17 different countries. So we're very excited to see the results of these clinical trials, which will be made available very soon. Hopefully we'll have some another tool here in the toolbox to fight COVID-19. So just to quickly segue to another paper, a very collaborative paper, as you can see, where we generated a protein-protein interaction map, not just to SARS-CoV-2, but SARS-CoV-1 and MERS, because we wanted to do cross-comparative studies. We also did localization experiments, combined it with CRISPR-based genetics, which I'm going to highlight here in a second, did also structural analysis, and also tried to connect this to real-world clinical data. So you can see there's 200 authors on this particular paper from 14 different institutions in 6 countries. I would say a work like this normally would take 6-7 years. It took about 4 or 5 months, and that's a testament to the collaborative spirit that went into this particular effort. So based on today's seminar here, I'm going to just jump to the CRISPR-based analysis that we did. So the genetic analysis, we took 332 interacting proteins, worked with Veronica and Marco there at the top at the Pasteur Institute, used RNAi to knock down these genes in A549 cells expressing ACE2. We used RT-PCR as our readout. In parallel, with the arrow going down to the bottom, we collaborated with Synthego, specifically Jared and Kevin, where they knocked out each one of these genes using CRISPR and COCK2 cells. We sent those cells to Chris Bassler. He did another assay for us where he used an antibody against the SARS-CoV-2 protein as the readout. So we had two different systems here that we're looking at. And then the bottom right, in total, out of the 332, we had 73 genes, at least in one of these systems, that had a statistically significant impact on HIV infection. Another way to look at this is in a network view. This is what I'm showing you here. The blue nodes are the 73 genes. The edges correspond to physical connections to the different coronaviral proteins. I want to zoom up on one part just in the middle here. It's a connection between ORF9b, a SARS-CoV-2 protein, and it exists in SARS-CoV-1, actually not in MERS, but we found it physically interacting with this protein TOM70, a mitochondrial protein. And we found that when you CRISPR out TOM70, you have an order of magnitude decrease of infection in COCK2 cells. So we had a number of lines of evidence suggesting that this particular connection was very important. And Klim Verba, who leads the QCRG structural biology group that Jacqueline told you about, the QBI fellow, he and his team got a cryo-EM structure of TOM70 binding to ORF9b. So ORF9b is in orange there, and TOM70 is in green. And TOM70 is doing a number of things, including regulating mitochondrial function and the immune response. We actually think it's the latter function that ORF9b is manipulating for the benefit for the virus. And we think that this is connected to a number of variants as well. Just on the right, there is some information about the structure. 36 out of the 38 ORF9b residues are interacting with TOM70. And there's a couple of phosphorylation sites that we identified on ORF9b that if we're phosphorylated, there's no way it could interact with TOM70. So we think phosphorylation is regulating this key interaction. And we're focusing a lot of attention on this interaction. And I love it because it shows you the value of combining proteomics and genetics and structural biology and virology to narrow in on a key node, which we think is important for SARS-CoV-1, SARS-CoV-2, and ultimately SARS-CoV-3. And we're doing a lot of fragment-based screening right now to find a compound that would interrupt this particular interaction. And as I alluded to before, we're generating a lot of information on these different variants. We think this is a key node that the alpha and the delta variant is using to better infect our cells. And we're going deeper into that. I won't talk to you about that, but I will say is that we're looking at proteomics, drug screening, CRISPR-based genetics, transcriptomic analysis, and also a work in bat cells to do a comparison across these different variants. And just very briefly, we use a similar approach across many disease areas. We had 3 papers that came out just a couple of weeks ago in Science. We're combining essentially proteomics and genetics, mostly CRISPR-based genetics, to get at insights behind a variety of cancers, including head and neck cancer and breast cancer, and working with Troy Eidecker, putting all this information together to come up with a hierarchical model of the cancer cell, which we're now using in combination with AI to make predictions about precision medicine. So we're very excited about this platform, and we're going across many different cancers and many other disease areas as well.
And so we wanted to tell you also a lot about the future plans we have for collaborations using the various platforms and technology that we have that Nevin's just gone through. And we're currently focusing on collaborations in Africa that we have. Mainly, we are starting with the University of Ghana, where we are working on a couple of cancer projects with them. But they have high sequencing capabilities, which we're going to be using along with eventually CRISPR and proteomics. And we are with all these countries that we're going to show you, we are assisting them and working with them to get mass specs into the area. We are also working with Uganda. I mentioned earlier our scholar, Jackie Lugemwa, who's there, who's currently the only person in the country with knowledge of mass spectrometry, because during her year at UCSF, she took a course. And so we are reinforcing that relationship there. And we're currently working with her on COVID and HIV and looking at their abilities for sequencing, as well as further down the road to be doing some knockouts and various things with cohorts and samples that they'll be sending our way. We are also working with Nigeria, where they have actually the capacity to house various machines, such as cryo-EMs or mass spectrometers, because they have an institute of note that is almost finished. And we are working with Christian Happy there on various viruses, Lassa, Ebola, Marburg, to name a few. And finally, we are working with our partner in Sierra Leone, by Laura Barry, who is currently also doing a Ph.D. in policy at UCSF, although he's already doctor of note in Sierra Leone, who opened a significant clinic on HIV, which was also a first responder to the Ebola outbreak there at the time. So we are looking at having a symposium in Sierra Leone this coming year. It's been postponed for 2 years now, which will be the first symposium on infectious diseases in Sierra Leone. And we invite any of you to reach out to us to be part of this. We are therefore looking at various future plans and breaking silos worldwide. We have a number of QBI scholars for women who have been lined up, but due to the pandemic held back, from Poland, Nigeria, Ghana, and other places. And we soon have a grant that we put in at NIH with 8 institutes in India and 6 in the United States for exchanges of trainees on infectious diseases such as tuberculosis. And the symposium, as I mentioned, sorry. And finally, we're planning on the psychiatry and neurology symposium with an institute in Argentina. And with this, we'll be wrapping up by telling you about a paper that Nevin and I were asked to put together for Cell to describe our work in creating collaborations around the world and what we use. And it's a bit of a how-to piece of how to involve young scientists, academia, industry, philanthropy, importantly, media and communications, and as well the use of symposia to create a dialogue around visions. Nevin?
Yeah, and just to add to what Jacqueline said, I think a lot of us would agree that there are problems with the scientific enterprise, academia and industry. And one of the major problems is that it's incredibly siloed, as Jacqueline alluded to, but it also rewards the individual. One person gets a grant, one person gets an award, one person gets tenure, and in doing so, you often discourage collaboration. But during this pandemic, it's been remarkable to see how fast we can all move together when we work together, when we break down these silos and don't worry so much about these individual awards. So I think the challenge now is to take what we've learned and apply it to the future. And the question now is, when the dust settles on this pandemic, are we going to keep this infrastructure and spirit alive so that we're better prepared for the next pandemic? And then the larger question is, why aren't we working together like this on all diseases, on breast cancer, on Alzheimer's, on Parkinson's, on heart disease? And I think because of this pandemic, which has obviously been a tragedy, there's a silver lining. One of the silver linings is in 20 years, we're going to look back and say, hey, what we learned during that pandemic, we applied to all these other disease areas, and we have so many more treatments, and we can help out so many more people. So that's the goal. And as Jacqueline said, her and I wrote up a piece describing what we did during the pandemic and how it can be used in the future. And actually Nature as well highlighted this collaborative effort. It was actually on the cover of Nature a couple of months ago. So if you're interested in these activities, what we've done and what we plan on doing, I'd encourage you to read these 2 pieces. And here's the last slide. This is a picture of our lab that was taken a couple months before the pandemic. There, obviously, a lot of people are involved in this, and just to highlight a few, the bottom left is Dave Gordon, the upper right is Gwendolyn Jang. They spearheaded our protein-protein interaction work on SARS-CoV-2. Mehdi at the back there is a postdoc. He's a computational expert. He helped out in all of this work. And then Robin Cake, Ruth Ittenhain, and Daniel Slaney, the many people circled, fantastic mass spectrometrists. They're crucial for all of this work that they described and all the work that we really do. So I'd like to thank them, and we'd like to thank all of you when we have.
Thank you.
Hey, great. Thank you both for such an informative talk. There are several questions in the chat. If you wouldn't mind taking a moment just to answer one before we move on. One of the audience members wants to know, have you thought about doing proximity labeling?
Yes, we have actually developed approaches in the past using Apex, the Apex tag. We are doing this. We're also BioID and TurboID to get at obviously the more transient interactions that could be important. We're also employing cross-linking mass spec approaches to get at those transient interactions. But the one thing that I think I'd maybe end with saying here is, which is kind of related to this question, one of the things that we're doing now is combining CRISPR, mass spec, and cryo-EM all together by tagging on the genome with CRISPR important factors that are connected, host factors connected to, say, SARS-CoV-2 or HIV, purifying them, plus and minus infection. You can do this poorly, doing mass spec, doing cross-linking mass spec, and going right to cryo-EM. Okay, and to me that is incredibly exciting to really look at how these complexes change during infection and combine all these different approaches together: CRISPR, proteomics, and cryo-EM. So it's not just CRISPR, it's not just mass spec. It's about putting it all together and combining it with chemical biology and structural biology. That's the vision here, and I think that's where the big breakthroughs are going to come in. One of our visions here, obviously, as Jacqueline alluded to, is to try to get some of these technologies in the hands of people where some of these diseases are incredibly problematic. So it's about connecting genes and proteins, but it's also about connecting people around the world.
All right, great. Thank you both so much. We'll move on now to the third talk, which is our second joint talk by Drs. Michael Ward and Mark Cookson. Dr. Cookson is a cell biologist whose current research efforts include the effects of mutations in genes associated with neurodegeneration at the cellular and molecular level. And Dr. Ward is a neurologist and cell biologist who studies the underlying causes of neurodegenerative diseases with an ultimate goal of developing targeted disease-modifying therapies for affected patients. So thank you both for talking today.
Great. Well, thank you for having us. It's a real pleasure to be part of this session. I really enjoyed that last talk about breaking down silos. And hopefully what we can do is convince you that we're taking a similar kind of approach, at least philosophically, for breaking down silos and enabling research within the space of Alzheimer's and related dementias using this project. So, as was said, I'm Mark Cookson. I'm a cell biologist by nature, and I'm co-leading this iPSC Neurodegenerative Disease Initiative, or INDI, with Michael, who's here from the sister institute, NINDS. And so let's talk about first, in the next slide, what are some of the issues that we know about with iPSC work? So I think one of the key issues, and we talk a lot about the issues around rigor and reproducibility of iPSC work, which is that for the most case, you would take a patient line, so a fibroblast or PBMC from a patient, make iPSCs, and then revert those mutations back to the wild type variant. And you know, of course, that works very well. You have then a pair of lines that you have some confidence we can compare. And there are collections of these in the second half of the slide here. Answer ALS, for example, has something like 1,500 lines collected from patients. NINDS has supported Rutgers to make a catalogue of disease-associated lines that should be available. But of course, there are difficulties with that, right? So the first is that the familial mutations, so those genetic variants that are of large effect size, are relatively rare and often hard to access. Making isogenic lines is not always done, and they're not often quality controlled. Just in the last talk about democratizing research, I mean, something that can get in the way is just legal agreements for sharing. And then I think most importantly for us, it's very difficult to make a comparison between, say, a presenilin mutation and a frontotemporal dementia mutation, because they're in different backgrounds from different cells, often made in different ways. And so our approach to doing this was to set up this iPSC Neurodegeneration Initiative, INDI. And the initial scope of this is to make about 150 or so variants in Alzheimer's and related dementias. So these are defined. So Alzheimer's disease, diffuse Lewy body disease or Lewy body dementia, frontotemporal dementia, which sits on the spectrum with ALS, and then make those all in consistent backgrounds. So the initial set will be the parental line, and I'll talk about that in a minute, both heterozygous and homozygous mutations, but then taking the pathogenic variant and reverting it back to wild type. So the real trio will be having a mutation introduced and a mutation reverted. We'll also make knockouts, some knock-ins for halo tag. And I will say at this point, and in interest of full disclosure, we had 3 external partners for this. So the Jackson Lab have been a major partner, but Synthago also helped us here, and Thermo made some of the lines as well. And we had some redundancy between the different suppliers there. Michael's going to talk a little bit about phase 2. So I'm just going to focus on the first phase of this, which is that engineering phase. How do we engineer iPSCs at scale? How do we generate all of these things in a relatively short order? So in the next slide. So I'm not going to, there's not a lot of data in this talk today. There's a lot of sort of conceptual things. This is probably the most complex thing that we did in the first year, which was to pick some lines that we're going to then use for engineering. And as you can see, we did a lot of quality control and decision making. Some of it was around genomics, so understanding what the genetic background of the lines were, understanding that they didn't carry known functional mutations, but also establishing that we can edit, genotype, differentiate in ways that we think the field would want to use, and that, of course, they retain pluripotency without any obvious problems. So in the next slide, what we'll show is the first 8 lines that we looked at. And again, the data here isn't particularly critical, but it shows you the things that were most important to us. Using the methods that we use, the methods that Synthego used, the editing actually wasn't really predictive of whether we're going to use a line or not. One of the key things was that the line grew well, but still had an intact P53 system. And so to the best of our knowledge, none of the lines had an oncogenic event. So the one that we've ended up using is a variant of a HIPC line called COLF2, which had a mutation but was then corrected and subcloned. So that's going to be called COLF 2.1. And for the first round of editing, that's the one that we'll use. And we have confirmed that it can indeed, differentiate, it grows well, it edits, and it has no pathogenic mutations. So the next slide. So I'm going to hand over in a minute, but I'm going to talk very briefly about what our plans are to distribute. So as you can all imagine, distribution of lines at this scale is really, there's not many people that can do it. We have committed to open science where we can, so the plasmids for this are all available on our gene already. We expect to be distributing a trio sets, so again, the parent, the mutant, the revertant early next year, and that will go through Jackson laboratory again. It'll be very similar to when you order a mouse, so you'll be able to just literally pick it from a web-based catalog. We have also started distributing few lines on an early access basis, and if anybody on this call wants to get access to them, they can contact myself and Michael, and we can tell you what's available, what's been made. We haven't necessarily done all the QC on this, so we're hoping that people can give us feedback and say that they, that it behaved well in their lines. And someone in the question is asking about regulatory consent. This line is, to the very best of our knowledge, free of any MTA issues. It can be shared under a very simple MTA, and we have the full provenance back to the original consent. So there's really very little in the way at this point of distributing this in a way that's completely compatible with legal and ethical constraints. So if we go next slide. I will say just briefly, so I mentioned that we started with Alzheimer's and related dementias, and that was because that's what we were funded for. And that was sort of a set of 150 genomic variants. But once we had the structure up and running, other people saw it and said, well, we'd like to add to it. So with aligning science across Parkinson's disease foundation, we've added about 20 so pure Parkinsonian variants, so without dementia. And through the Chan Zuckerberg folks, we've added 16 neurodevelopmental variants that were not in the initial set. So part of what we're trying to say is, as well as being open, we're also making something that scales well and can be added to. And in fact, as you see here, there's an effort to make really functional lines, so things like DEDCAS9 and the like. So things that can be then used as tools, but again, in the same genomic backgrounds. So I think with that, one more thing on this early access. So on measure of success here, I was really interested to talk about measures of success in papers in the last talk. Our measure of success here is that people use the lines. That's the key thing. And we've made 150 projects approximately so far, and those are out in 50 labs in 10 countries across 3 continents with no limitations. So, you know, I think this is what success looks like for one of these democratized projects. So with that, I'm going to hand over to Michael, who's going to give you some, you know, really specific examples of how this is working for us.
Great. Thanks so much, Mark. So I'm going to start off here by just telling you about 3 vignettes about some of these users of the early access lines, because this is just something that Mark and I are very proud of, that people are already starting to adopt these and use them in their own research. And then tell you a little bit about phase 2 of INDI, which is really this foundational data generation of phenotypes that ensue in disease-relevant cell types generated from iPSCs that harbor these mutations. So the first vignette is from Gujan Boo's lab at Mayo Clinic, and they're very interested in ApoE4 biology. So they've requested the entire suite of ApoE mutant lines and performed karyotype analysis. It was all normal, and markers of pluripotency, which were again all consistent with a pluripotent iPSC line, differentiated these successfully into 3 germ layers, and they're now well on their way toward generating organoid models of Alzheimer's disease with these different ApoE4 alleles or ApoE alleles. Next set would be from Erica Holzbauer's lab at University of Pennsylvania. They're really interested in vesicular transport, in particular neuronal autophagy, which is quite a bit different from autophagy that goes on in other cell models. They differentiated COLF 2.1 iPSC line into neurons with the neurogenin system, and then they performed time-lapse microscopy of retrograde moving autophagosomes here. And actually, it was really neat because they looked at the control parental line and then a LRRK2 mutant line. And you can see here in this chymograph of this normal, gradual retrograde transport of LC3 positive autophagosomes in the control neurons. But in this LRRK2 mutant, you can see how these autophagosomes just freeze in place. This is a phenotype that had been reported in a different iPSC mutant line with this same mutation. So it was really gratifying to see that they could replicate this exact result in the knock-in line from the INDI set. And this was work that was done by Dan Duo and Erica's lab. And then the third set, and I just think this experiment is really, really cool. It is performed by Deborah Cronenberg-Versteg. And she has this elegant model in which she takes iPSCs, human iPSCs, differentiates them into microglia, then depletes microglia from organotypic brain culture, brain slices, either from mice or from humans, from intraoperative specimens obtained from human patients. Gets rid of the endogenous microglia, and then she puts these iPSC-derived microglia on top of the slices, and what happens is those iPSC microglia invade, because there's no resident microglia anymore. She wiped them out with antibodies, and they ramify gorgeously. To me, somebody who's looked a lot at microglia, I've never seen any iPSC microglia that look this beautiful. And this is being done with the COLF 2.1 line in these human brain slice co-culture. So what we've seen and heard from investigators who've requested these is that it looks like the COLF 2.1 line is going to work for a lot of different applications, differentiating into neurons, into glia, making organoids. I think this line is going to be a workhorse for the field. So we really couldn't be happier about how the community is adopting this. But in the last few minutes, I want to tell you about something that's really exciting that's happening on the NIH campus. And I think actually, in many ways, is a parallel to a lot of the fantastic work that Nevin Krogen just described about what's happening at UCSF and Gladstone. And that is, how do we start to understand ramifications of these disease-related mutations on biology, and do this in a scalable manner, and do this in a multimodal fashion, where you're not just looking at one type of data, but you're looking at a concert of different kinds of data sets that play well together. So what we've done is we've begun to develop a series of technologies that will let us differentiate these different mutant iPSC lines at scale. This is a completely roboticized process. It's being carried out by a Hamilton robot, which is set to arrive in the new building for CARD in February. We're going to be performing differentiation of these iPSCs at 48 batches at a time into neurons, and then followed by a set of high content microscopy, functional synthetic lethality CRISPR-I screens, total RNA-seq, and proteomic mapping, not just of the proteome of the cells that harbor these mutations, but also APMS-based strategy with halo knock-in lines of wild type and mutant proteins. Once these data sets are generated and validated with QC metrics, they're going to be uploaded into an open data repository on the fly, searchable by the field. And we're going to build this data repository in such a way that not only can people explore the data that's in there, but they can use this data as cross-referencing data sets against their own data sets that they generate in their lab. So we're very excited about this project. I'm going to show you some of the tools that we've been building that I think is going to enable this. First of all, here's just an image of what this robot's going to look like that's going to do the automated cell culture. Quite similar in many respects to the Hamilton robots that the New York Stem Cell Foundation has successfully used in their own high-content iPSC culture. We've modified a version of our neurogenin differentiation protocol that can be actually transfected into cells with an automated lipofectamine stem technique. It's a piggyback-mediated transposon that can integrate TET-on neurogenin as well as selectable markers. And this allows for a robot to generate these things en masse. It doesn't require safe harbor engineering and picking clones. This is something that can be done in a polyclonal population. So really amenable to high content approaches. This is an image of the Colt 2.1 line with the piggyback neurogenin differentiated from iPSCs into glutamatergic neurons. We're developing these protocols as we speak and refining them. These are going to be accessible to the community in protocols.io, and actually we should be releasing this one for the piggyback version pretty soon. All the plasmids, as Mark mentioned, are being deposited onto AdGene. So these neurogenin neurons from Colt 2.1 map, as we would expect, to fetal-aged excitatory neurons shown in these UMAP projections here. This is done by single-cell sequencing. So we think we're making relevant cells. They're not aged cells, but then again, this is iPSC cultures. We don't expect that. And then we've invested a lot into automated proteomic workflows here. A lot of the stuff that groups do is easy to do when you're doing one set of proteomics experiments, but once you start to scale this up and you're pumping out samples from a robot, you need a different approach. So actually, this is a lot of help from Nevin Krogen's group in terms of getting an automated SP3 protein extraction technique up and running on a Kingfisher instrument. Now a human won't need to touch the samples as they roll off the robot, go directly onto this Kingfisher instrument for SP3 protein extraction. And this goes into thermo scientific eclipse mass spectrometers with FAMES Pro and data independent acquisition mass spec. These methods look like they're revealing far more peptides than the more traditional methods that we were using previously here, just with whole cell proteomics. And you can actually look and see as iPSCs differentiate into neurons and their complement of proteins. We're going to be using day 28 iPSC neurons. And the traditional method by hand, you can see the spread on a PCA plot here across samples. It's much tighter with this fully automated Kingfisher SP3 method that we're going to employ at scale. And then the next is something I'm particularly excited about. This was really work that grew out of a collaboration with Martin Kampman at UCSF that enabled us to, for the first time, do genome-wide CRISPR inhibition screens in iPSC-derived glutamatergic neurons, the same neurons that we're going to be making in Indy Phase 2. One takes these iPSCs that we introduced neurogenin and DCAS9 technology with piggyback, then overlays these with a genome-wide guide library, differentiates these into neurons, and basically counts guides at the beginning and the end of the experiment to find out the effect of gene knockdown on neuronal fitness. When you do this in a test case by knocking down a neurodegeneration-related gene TP43, one can actually then interrogate buffering genes and synergistic lethal genes, very, very similar to the synthetic lethality screens that were done in yeast very successfully, but in this case in disease-relevant iPSC-derived neurons. Now, this initial technology wasn't really scalable for a robot. It required 100,000 different guide RNAs in this second-gen library. So we've gotten access to a third-gen library from Jonathan Weissman that's reduced the size of the library from 100,000 guides to get genome-wide coverage down to 20,000 guides. So this lets a whole genome screen with 1000x coverage being done in 20 million cells. That's something that can be done on a single 15-centimeter dish. And so when we look at the performance of this single guide library versus dual guide library, we actually find that in the neurons, the dual guide library is outperforming the single guide library, despite the fact that it's 1/5 the size. You're getting larger effect sizes for each targeted gene with this particular library. So I think that now we're at the scale of being able to do this genome-wide fashion with automated robotic production of the neurons. And then finally, this high content microscopy. We're using a super res Sora spinning disk with an auto plate loader. This is very similar to what Steve Finkbinder is doing at Gladstone. We're applying a cell painter type approach that Ann Carpenter pioneered at the Broad. Here's just some images of mitochondria from these Col 2.1 neurogenin neurons here. We can make out the very fine detailed structure of these organelles. So this is going to be a multi-organelle high content imaging platform to generate data for artificial intelligence interrogation. So with that, I'd like to thank the team. It's really been amazing. Andy Singleton is the leader of the Center for Alzheimer's and Related Dementias, who really championed this project and allowed us to get the necessary funding to launch this. Having deep collaborations with a number of other groups at the NIH, which includes Data Technica International, led by Mike Knowles. For us, Fagri has been really intimately involved in a lot of the CRISPR-I work here. Liron Peng in the single cell work, and then an amazing team that we've assembled for India itself. Erica Flores has been doing all the cell culture, Andy Key leading all the mass spec work, Dan Ramos doing the functional genomics, and then Marinita, who actually isn't on the slide, but leading the high content microscopy. All of the engineering was done by Synthego partners as well as JAX. The JAX team led by Bill Scarns, just one of the most delightful people I've ever collaborated with. And then CZI has really been an essential partner here. Mark Sutherland, Katja Rose, and Alden Connor. We've had great input from collaborators in Cambridge, Florian Merkel, Andrea Yang, and actually a lot of the nuts and bolts of this project were worked out with a long-term collaborator and colleague of mine, Bruce Conklin at Gladstone. So with that, if we have any time, happy to answer any questions.
Okay, great. Thank you both so much. From the best I could tell, Dr. Cookson was answering the questions as we went along in the chat, I think. And since we're about 10 minutes over, if it's okay with you all, we'll just move on to the next talk for now. And perhaps you could answer more in the chat. Sure. Okay. Great. So thank you again for that talk. And our next talk is from Dr. Luke Dow. He's an associate professor of biochemistry and medicine at Cornell Medicine in New York City. His lab is focused on understanding the biology of colorectal, pancreatic, and lung cancers and developing new therapeutic approaches to treat these diseases. Thank you, Dr. Dow, for talking today.
All right, thank you. You can see the slides okay?
Yes.
All right, excellent. So thanks for Synthego for the invitation to speak. What I want to talk about today is our effort to use base editing to try and understand some of the complexities underlying cancer initiation, progression, and also therapeutic response. A lot of what I'll talk about will mirror some of the concepts that Emanuela opened the session with. So we'll get into that in a little bit. I think the problem that we're really trying to address is at two levels. The first is this, that mutation patterns in cancer are extremely complex. This is an oncoprint of the top 25 most mutated genes in colorectal cancer, where each column here is an individual patient's tumor. And you can see, obviously, that no individual tumor carries the same combination of mutations, and also that the types of mutations that occur in each of these cancers in each gene are very different, as indicated by the colors. The second level of complexity that we're trying to understand is that within individual genes, the mutation patterns are also complex. Now, these are the three most commonly mutated genes in colorectal cancer. APC occurs in over 80% of sporadic and familial colon cancers. P53 is very common, particularly in advanced disease, and KRAS is almost in 50% of cancers. I show these three examples not just because they're common, because they represent three different kinds of mutation patterns. APC is a classic tumor suppressor. We see lots of nonsense truncating mutations and very few missense alterations. P53, we see a wide variety of missense mutations with multiple hotspot mutations where we see piling up of individual mutations throughout the coding sequence. And KRAS is a classic oncogene where we see very high accumulation of hotspot mutations, particularly at codons 12 and codons 13. Now, we have anecdotal data from our lab as well as many other labs around the world that the types of mutations that you see in these genes not only have an impact on the way a tumor develops or whether a tumor develops at all, but also the way they respond to particularly molecularly targeted therapies. We've seen this in APC, many examples in P53, and more recently through the development of new mouse models in KRAS. So the the anecdotal examples are great, but how do we ramp up and understand the enormous amount of complexity that we're dealing with in individual cancers and across the population? How do we do this at scale? Now, fortunately, from a modeling perspective, the mutations that we see in cancers are not completely random. Now, obviously, we get all different types of mutations, and these are due to underlying mutation signatures, some of which are not completely understood. But there is a significant piling up of mutations that are C to T transitions. Also, fortunately, we now, as of about 5 years ago, have tools to be able to try and mimic these mutations in the dish. Now, this, I'm sure most of the people on the webinar are aware, is a CRISPR-based base editor. This was pioneered by Alexis Comer and David Liu's group in 2016, described the first cytosine-based editor that incorporated this ApoBec domain that allows you to, within a small window of the guide targeting region, engineer specific C to T transitions and create single nucleotide variants within genes designated by the targeting by the guide RNA. So we wanted to use these tools to try and understand cancer variants, not one at a time, but at scale. So we took MSK impact data of about 20,000 different tumors, pan-cancer, and looked at mutations that occurred across all these cases. And this encompasses about 460 cancer-associated genes at the time of the data collection. Now, we used an input file and built a computational pipeline that would allow us to put the mutation file in and specify specific Cas and base editor enzyme properties, and then predict both in the human genome and the mouse genome which mutations could theoretically be modeled by cytosine base editors. And overall, this allowed us to build design predictions for human and mouse of about half of the mutations that are seen, again, pan-cancer across this data set, encompassing missense, nonsense, and splice site mutations. In the mouse, the mutation number, as well as the number of guide RNAs that could theoretically make these mutations, is smaller due to changes in codon usage at the specific sites of these mutations occur in humans. Now, rather than designing this library like a traditional CRISPR pooled library with guide RNAs, we wanted to incorporate an additional feature which others have used often to build computational prediction pipelines. We call this a sensor-coupled library, whereby the sgRNA is flanked downstream by its cognate genomic target with genomic sequences either side of that target. And so when the sgRNA is expressed and an editor is provided, it can target the same piece of DNA, and you can amplify this entire thing by PCR and sequence it and get a readout of the activity of the sgRNA rather than directly sequencing the target site. So when we do this with multiple different enzymes, we see that we recapitulate many of the features that we know about base editors. This plot shows the percentage of C to T editing across the window of the cytosine or the protospacer, going back 5 bases and all the way through the protospacer. And we see high levels of activity between about positions 3 and positions 9 of the base editor in these canonical enzymes. Here's one that was engineered in our lab called FNLS. We see in enzymes that have expanded windows, these extend out to about positions 12 or 13 of the protospacer. And this is not true of any given cytosine in the window. We see there's a strong dependence on the dinucleotide context, or the nucleotide that immediately precedes the target cytosine, where TC dinucleotides are edited well across a wide range of the protospacer, whereas GC dinucleotides are not. And much of this has been described previously as well. We see, using this sensor-based approach, expected features of targeting, whereby using canonical enzymes, we target sites that carry NGG PAMs, less so with NAG and NGA, unless you use more PAM-flexible enzymes like the NG Cas enzyme, where you can less efficiently overall, but more broadly target these PAM sites. Of course, none of this is really useful at all unless the editing site at the sensor recapitulates what's happening at the endogenous locus. And we took a number of examples from our data set and went and one by one tried to engineer these same mutations at the endogenous genomic site. This is data from PC9 cells. And you can see in most cases, in more than 50% of these cases, the efficiency of editing at the sensor site is within about 10% of the endogenous site. And those that are not, more often than not, show higher levels of editing at the endogenous site than at the census site. So overall, we feel like we're predicting pretty accurately the efficiency of these guides, and certainly within a number of guides that are targeting the same region, the ranking of those guides, while maybe different cell line to cell line, stays pretty consistent. To make this data useful to a number of people, as I said, we've scanned now maybe 1,400 different mutations with over 6,000 guides. We wanted to make this broadly useful for the community. So we built a web app that contains both sensor-validated guides where you can go in and select specific attributes that you're looking at, so the species that you want to design guides against, the gene, whether you have a bias for a particular enzyme, whether you want to see a particular mutation, and what kind of cell line we may have done it in, and output these tables that have editing efficiencies. We also have a separate tab on this, which is a purely design tool. So these have not been sensor validated, but it's an expanded prediction set using a much larger range of Cas9 enzymes and base editing configurations, some of which don't even exist yet. But given the speed of development of these tools, we predict will exist in the near future. In this example, just showing, looking for one specific gene, p53, and there's more than 1,500 potential guides to engineer p53 mutations. The website also contains a lot of information about features of base editing enzymes. So the website address for this is down below, and we hope this will be useful to people to either design their own sensor libraries or clone individual guide RNAs. So one of the things we hope this will enable is for people to just go and take guide RNA sequences off the shelf and go straight to do their important experiment rather than doing months and months of pre-validation. This is one example of doing this, where we selected a range of p53 targeting missense mutations across the p53 coding sequence. We selected these particular ones because from the sensor data, they showed a high level of editing purity. They had sort of medium levels of actual cytosine editing, but the editing purity was very good, meaning it hit the target cytosine only and not many cytosines around the window. We could then take these guide RNAs, introduce them into pancreatic ductal epithelial cells that are p53 wild type but contain an oncogenic KRAS mutation, transplant them without selection into the pancreas of a mouse, and ask whether tumors develop. And in each case, from these pre-validated or sensor-validated guide RNAs, we got tumor development, often reflecting the editing activity of the guide, because I mentioned these were not selected prior to implant. But when you look at the tumors that come out of these, look at the cells that come out of these tumors, they have very high levels of editing activity. And these are mixed populations. This is bulk tumor, so it contains immune cells and stromal cells. One of the other uses of these tools is actually to use the sensor library itself to do functional screens. So in this case, we use the same cells. These are PDEX cells expressing the FNLS base editor. We can introduce the mouse base editing library and then perform any kind of functional screen. In this case, we did the most simplistic screen you could imagine, which was just to put the library in and allow the cells to proliferate. So there's no real selective pressure other than proliferation in the dish. And then you get two types of information out of these screens. You can calculate sgRNA enrichment, so whether you had low fitness variants or high fitness variants, in this case, proliferation, as well as whether you saw editing at the surrogate sensor site, so whether the guide RNA was actually active. And then you can use both of these metrics to ultimately prioritize hits. Now, if we do this screen, you can see there's some depletion of some guides, but a very small number of guides show very high levels of enrichment. These happen to be, as you would expect, p53 guide RNAs. And if you focus just on the p53 guide RNAs that are in the library, you can see the ones that are enriched here are the ones that show high levels of C to T editing. So you already have some pre-validation that the hits you're getting in the screen are not really background noise, but are the ones that are causing editing. You can look at this data in another way by plotting the log fold change from day 1 to day 30 in this case, and then looking at how that log fold change compares to the editing you see in the sensor. And this data, obviously, we see a lot of p53 guides. There are a number of other hits in here which we're following up. You also start to see guides that showed high levels of editing in the sensor but don't really show functional enrichment. And so these would be potential candidates to say these are p53 mutations that don't have a functional impact on proliferation in this context. When you categorize this using the OncoKB classifier from MSKCC, you see that When we start with the guide RNAs, we have a number of known or predicted oncogenic mutations. After the screen is complete, those that don't show enrichment see a slight decrease in the known and oncogenic prediction criteria. The ones that do show log-fold enrichment have a slight enrichment of those known or oncogenic predicted mutations. But if you incorporate both the editing level and the log-fold change, you get a much more dramatic enrichment, and more than 70% of these mutations are either already known or predicted to be oncogenic. So we have a large data set now, and I hope people will be able to use the web tool there. We also need systems to be able to ask questions about cancer development in organisms, in complex cultures ex vivo. And to do this, we've been developing a mouse tool, which we're calling IBE, which is an inducible and reversible base editing mouse. And we're using an optimized base editor that we developed a few years ago, and this is through a tetracycline inducible system in the mouse that we've used for many other applications in the past. The first steps to try and check that this mouse was working effectively was to look at activity of the base editor across multiple different organ types. As mentioned in the beginning, these are the 3 organ types that we're particularly interested in the lab, and we've been making organoids derived from these tissues, from these mice, to check editing. We did a first pass to look at editing activity using a fluorescent reporter that we published last year now, that expresses a constitutively nuclear localized scarlet, and then an inducible GFP, whereby cytosine editing creates the ATG start codon of GFP and allows GFP to come on. So if you look in this pancreas here, not treated with DOX, you see constitutive scarlet, but no GFP. But just 2 days after DOX induction, we see GFP now expressed. And the same is true in these DOX-treated small intestine and basosphere-derived organoids. I don't want to spend too much time on this, but there's been a concern going the last few years that a high-level induction of base editors can have really deleterious effects on cells, both at the sort of transient RNA editing level or the DNA editing level. We wanted to look at this because obviously we're trying to build cancer models. We don't want to have these collateral effects. And so we took organoids here with the Mugus reporter. We activated with DOX for either 4 days or 8 days, and then switched DOX off at day 4 and looked at RNA editing. And to cut a long story short, we didn't see any evidence of RNA editing, whether the base editor was being expressed in these cells or not. And I think this is really due to the fact that we have a single copy integration of the enzyme. We're not getting outrageous levels of the base editor. And this is actually consistent with some of the data that's already published on single cell analysis of RNA editing, where low expressions of the editor didn't cause RNA off target effects. So we've been using this in organoids. We really would love to be able to genetically manipulate organoids and do this in multiple different configurations, do multiplexed editing. When we don't add DOX to these cells and we transfect them with either plasmid or synthetic guide RNA received from Synthego, we don't see any editing. So there's very little leakiness of the base editor. When we transfect these cells on DOX, we see some editing with plasmid DNA, although in most cases it's actually very low. With the synthetic guide RNAs, we've been able to get upwards of 90% editing activity with particular guides, whereas others may be around 50%, but still very usable. I don't have the data to show you right now. We're waiting on more sequencing, but you can multiplex this. We've gone up to about 4 guides now, where you see efficient editing at each of those genomic loci with 4 different guides. So you can also do this in vivo. Here we're using a liver model where we can deliver a MYC transgene on a Sleeping Beauty cassette, as well as sensor-validated or sensor-predicted guide RNAs by hydrodynamic tail vein injection. And when you do this in either with single sgRNAs that cause liver cancer-associated mutations, like hotspot mutations in beta-catenin, or with just naked synthetic sgRNA from Synthego, you can develop tumor models in vivo using this mouse. So I'll finish there. And I just want to thank the people involved. Bianca Diaz and Ted Kassenhuber and Francisco Sanchez Rivera really spearheaded the base editing sensor library validation. And Alina Caddy has been driving the inducible base editing mouse. And just a quick plug for Francisco, who's heading off to start his own lab at MIT in a couple of months. I'm sure he'd love to hear from you if you're interested in continuing some of this work.
Oh, thank you so much for that talk, Dr. Dow. There are a lot of questions for you in the chat. I'm just going to pick one out if we can get through it kind of quickly. One of the audience members wants to know, what are the ramifications of your C-to-T analyses on current approaches to next-gen sequencing and therapeutic approach in cancer?
Ooh, I'm not sure how to answer that.
Okay, maybe we can take that one to the chat. It's kind of a big question,
Yeah.
a lot of implications. Okay. Well, a simpler question then, how long do you follow the mice in these studies?
Yeah, the mice, it's really dependent on the tumor model. So in the liver model, they can get sick anywhere from 4 weeks to 12 weeks. And that can be due to the level of editing or the efficiency of the guide or the transgenes that you deliver. So if you deliver combinations, they often come down faster than with individual guide RNAs.
Okay, right, great. Well, thank you again for that talk. And we'll move into Dr. Raffaella Soldi's talk. So Raffaella is a research assistant professor in the Applied Cancer Research and Drug Discovery Division, and her research focuses on translational and preclinical studies on human cancer with the ultimate goal of developing molecularly targeted therapeutic strategies to treat human malignancies. So thank you, Dr. Soldi.
Thank you. Hope you can hear me.
Yes, we can.
Okay, so yes, thank you for inviting me here to present our study that uses the platform of CRISPR to identify new therapeutic approach for cancer treatment. In particular, today we are going to talk about the Ewing Sarcoma. Ewing Sarcoma is a very rare and very aggressive pediatric cancer that target the bone and the soft tissue associated to the bone. And typically the survival rate of this cancer is around 60% if caught in early stage. Otherwise, if it's metastasized, the survival rate drop down to approximately 20%. And the only treatment currently in use right now is the surgery, so amputation, followed by chemotherapy, radiotherapy. So there is not a target therapy for this cancer. The Ewing sarcoma is driven by a chromosomal translocation of the gene EWSR and the ETS gene family member FLY1. It's form a chimera, EWS fly, which is a transcription factor. And unfortunately, obviously the first approach to when you want to develop a drug for treatment of this cancer will be targeted the EWS fly, but it's very difficult to target a transcriptional protein, and so we for two major reasons. One is because transcription factor per se is very complicated due to the high amount of interaction with different pathways. And second, more related to EWS fly, is because the translocation doesn't occur always in the same place. And so it will be very difficult and challenging to develop drugs that target the chimera and not the EWS or fly wild type. So we look for other option, and since EWS fly is a transcription factor, this protein will interact with epigenetic complex. In particular, EWS fly interacts with the complex NERD LSD1 to regulate the expression of genes that are tumor suppressor. And so approximately 10 years ago in our lab, in Dr. Sharma lab at the Huntsman Cancer Institute at the time, we started to develop a drug that was targeting member of this complex, particularly the LSD1, the lysine-specific demethylase 1 that promoted the demethylation of the histone 3 in lysine 4 and 9. We targeted this member because in our previous study, we showed that the downregulation of LSD1 results in a totally subversion of the genetic signature of EWS fly. And so LSD1 play a very crucial role in the transcriptional activity of EWS fly. And actually, we were able to develop a new drug, which is SP2577, which is a potent reversible LSD1 inhibitor, and is currently licensed to a pharmaceutical company and is in phase 1 clinical trial for Ewing sarcoma as well as other solid tumor. However, when we were developing this drug, we realized that this drug has a more cytostatic effect than cytotoxic effect, and most probably will work better in cancer treatment if associated to another drug to enhance the cytotoxic effect. And this is actually the aim of this study that I'm going to present today. And to identify genes that can be modulated by drug, resulting in downregulation of those genes, that together with the treatment of SP2577 promote the death of the cells, we use the CRISPR approach. By using the Synthego library whole exome CRISPR, we apply the target deconvolution approach in which you treat the cells with a drug and knock down the genes by one to identify genes that the knockdown lead to death of the cells upon treatment with SP2577. So for this study, we use only a portion of the whole exome CRISPR library from Synthego. We use the druggable portion, which are approximately 8,000 genes that cover genes that are either directly targeted by drugs or that are part of pathway that can be modulated by drugs. On the top left of this slide, you can see our workflow, which is extremely simple. We just transfected the Ewing sarcoma cell lines with the Cas9 and sgRNA from this portion of the library by using the reverse transfection. And then 24-hour post-transfection, we treat the cells with SP2577 at a concentration of the drug that is around the IC50, because the goal was not to promote that, but to see the combination of the drug plus the downregulation of genes if result in death of the Ewing sarcoma cells. This screen led to the identification of 103 genes that were either directly target of drugs or part of pathway that were targeted of drugs. But among those, only 19 were targeted by drugs that were FDA approved. And since the goal of this study was to actually find immediately something that can be used in clinical trial with SP2577, we decided to focus only on the genes that were modulated by FDA-approved drugs. So from 103 genes, we went down to 19. And among those, we focus on ABL2 because of its interesting characteristic in a tumor setting. ABL2 is part of the ABL family, which is a subfamily of the SRC family. Those are proto-oncogenic non-receptor tyrosine kinase protein that they share a striking structural similarity with SRC. And they are involved in a variety of biologic function, which include proliferation, migration, differentiation, and also survival of the cells. In a tumor setting, the ABL protein are, if we look at hematological kind of tumor, they carry mutation. The deregulation of those proteins is due to mutation of the ABL's protein. In solid tumor, instead, they don't carry usually mutation, but the deregulation of their activity is associated to an overexpression of those proteins. Among the ABL family, there are actually only two member, ABL1 and ABL2. ABL1 is the most studied. However, ABL2 play an overlapping role in the process associated to the survival and proliferation of the tumor cells. So the first things that we decided to do was actually validate our CRISPR finding. And so we knocked down ABL2 in different Ewing sarcoma cell line by using alternative methods to the CRISPR, so by using shRNA or siRNA. And in all cases, we were able to obtain approximately 50% downregulation of the expression of this protein. And then we were using the knockdown cells in comparison to the wild-type cells to see if first the knockdown was interfering with the proliferation rate of the cells, and second, if it was helping, like we saw for the CRISPR screening, the cytotoxic effect of sp2577. So here in these slides, you can clearly see that in all cases, on all the cell line that we tested, the knockdown ABL2 doesn't really affect the proliferation of the cells. However, when we look at the efficacy of sp2577 on those cells, you see that the drug is more cytotoxic when the cells have an ABL2 knockdown. And this difference between the wild type and knockdown is actually significant in all cases. We also perform anchorage-independent grow assay to verify if the knockdown ABL2 were still tumorogenic kind of cells. And so when as you can see here from this experiment, actually the knocking down ABL2 significantly reduced the tumorogenicity of the cells. And again, when we treat with the sp2577, also in this assay, you can see that the cytotoxicity of the drug is increased significantly. So how the knocking down of ABL2 help to increase the cytotoxicity of sp2577? So like I said at the beginning, ABL protein are involved in a variety of pathway in the cells. So we decide to go through all these pathway and see which one is or which pathway are involved in the regulation of an enhancement of the cytotoxicity of this drug. So the first pathway that actually we look at was the proliferation because of the results that I just showed you. But unfortunately, as you can see from these slides, the results are very inconsistent. So we are still working on that. So if it's not the proliferation, the next pathway that we tested was actually the apoptosis. So what pathway leads to apoptosis? And to verify that, we perform the assay to evaluate the level of cleaved caspase-3 in the cells that were ABL2 knockdown wild type after treatment with sp2577. When we performed this assay, actually, we were actually surprised to notice that when we treat the wild type cells with sp2577 for 24 hours at a concentration that should actually lead to the death of the cells, what we see is that there is a reduction of the expression of cleaved caspase-3. This doesn't occur when we knock down ABL2, and when we compare, we actually combine the knockdown ABL2 with sp2577, we recover the expression of cleaved caspase-3 as well as actually is increased significantly. And so we question maybe the ABL2 affect the apoptosis in Ewing sarcoma cells treated with sp2577. There are actually studies that show that in gastric cancer, the downregulation of ABL2 lead to apoptosis. And there are studies that seem to suggest that this mechanism may be associated to SRC downregulation. ABL2 and SRC has a very cross interaction. They activate each other. And so we question whether this low this apoptotic effect of the downregulated ABL2 cells treated with sp2577 was due to a downregulation of SARC. To do that, to verify that, we performed an experiment that is shown here on the right side of the slides, in which we treat the scramble cell, so the wild type cell line, Ewing Sarcoma cell line with sp2577 for up to 24 hours. And in parallel, we treat the same cells with a serum starvation assay to verify the downregulation of SARC. And also we perform a similar experiment on the cells that were knocked down for ABL2. So as you can see from this slide, the level upon serum starvation, the level of SARC goes down and is maintained for all the duration of the experiment. And this will lead to the apoptosis of the cells. But when we treat with sp2577, we have an initial downregulation of the activation status of SARC. That lasts at least for 6 hours. But then at 16 hours already, you can see that the phosphorylation is back. And at 24 hours, which is essentially this, the time point that is shown on the graph on the left side, the phosphorylation of SARC is actually higher than even the control. This doesn't happen when we have the knockdown ABL2. In fact, you can see that upon 24 hours, the phosphorylation of SARC is down. And when we combine sp2577 with the knockdown ABL2, after 24-hour incubation, the phosphorylation of SARC is still down. So this experiment seems to suggest that the enhancement of the cytotoxic effect of sp2577 in Ewing sarcoma may be due to the downregulation of ABL2, which result in downregulation of SARC and then promotion of apoptosis. The next thing that we did was to validate the efficacy on knockdown of ABL2 in vivo setting together with the treatment with sp2577. So we injected the nude mice with Ewing sarcoma cell line that were either wild type or knockdown for ABL2. And then after the tumor reached the size of around 200 millimeter cubic, we started to treat with sp2577 at a concentration of 50 milligrams twice a day. 50 milligrams per kilo. So this is pretty conventional for this drug. So as you can see from the graph on the right side of the slides, the wild type cells, obviously, the tumor grow as normal, as expected. When we look at the knockdown ABL2, actually, you can see that there is a huge increase of the tumor size. And then when we treat with sp2577 alone, we don't really see any efficacy in vivo. This is actually, unfortunately, what we already knew from previous study. When we combine actually the ABL2 knockdown with the treatment with sp2577, we have a reduction of tumor volume up to 50%, which is very significant. And here on the left side, I just show the weight that show that there is not actually toxicity for the mice for this treatment. So these are results. Those results are actually very exciting for us because, like I said, the drug per se is not really very cytotoxic, but in combination with all these experiments seems to suggest that the combination of knockdown ABL2 with the treatment sp2577 will enhance in a clinical setting the results for the patient with Ewing sarcoma. So what drug we will need to use in a clinical setting? So like I said before, the ABL protein are a subfamily of the SARC protein. So most of the drugs that affect the the function of SARC will affect also ABL2. Drugs like dasatinib and imatinib have shown that they bind actually ABL2 even with less affinity than SARC. And so we tested those drugs together with a new FDA-approved drug that is supposedly specific for the ABL family, asciminib. So by itself, those drugs, they don't show a very strong efficacy on the Ewing sarcoma cell line. Here we test on 2 Ewing sarcoma cell line because you can see that the IC50 is over 10 micromolar, while SP2577, which we know that works very well in 2D culture, is around 200 nanomole. But when we combine SP2577 with either dasatinib, as I show here, or imatinib, I don't show for reason of time, we have a very high level of synergy that is measured by Bliss synergy or Chow-Talalai that show that different ratio actually we have a nice synergy for the combination in both cell line. We also briefly test the asimentinib, sorry, I cannot see the name of this drug, but the drug that is specific for the ABL proteins. And you can see that also in this case, we have synergy islands present measured by the Bliss synergy. And also the Chow-Talalai is able to identify 2 ratios in which this combination of drugs can work. So in conclusion, we can say that the CRISPR screening on the Ewing sarcoma cell line identify a set of genes in which we focus on ABL2 as a possible target for drug to enhance the cytotoxic effect of SP2577. It's possible that the low level of ABL2 may promote apoptosis in the cells that are treated with SP2577 through downregulation of SARC. This is something that we have to study a little bit more in detail. But our results suggest that a combination of SP2577 with drugs that target ABL's protein may effectively suppress the tumor growth in a patient. And so also this study provide an insight on the role of the array CRISPR screening to identify or develop new therapeutic treatment for cancer. Thank you for your attention. If you have any question, I'm here to answer.
Thank you so much, Raffaella. That was a really great talk, and there are a lot of questions for you over in the chat. Since we're already over on time, if it's okay with you, I'll just move into our final talk of the session.
Sure.
Okay,
Thank
great. Sure, just let me get my slides pulled up. So I'm going to attempt to give a really quick talk since I know that we're already over on time. Camera going. Okay. Hopefully you can see and hear everything. Like I said, I'm trying to be mindful of your time, so I'll go a little quicker than I normally would. I'm Laura Lambert. I'm an assistant professor at the University of Alabama at Birmingham, and I'm the director of our transgenic facility there as of this month. So that's exciting. And at UAB, we're doing a lot toward unraveling these variants of uncertain significance. So that'll be the focus of my talk today. We have several different centers and facilities in place for examining these, and we have a lot of really great infrastructure. So today I'll talk about the interplay of 4 of our different facilities that I'm a part of, including our neurofibromatosis program, the transgenic facility, or transgenic and genetically engineered models core facility, is our official name, and then our newest center, which is our Center for Precision Animal Modeling, that we're really excited about. And we also have a lot of interplay with the Precision Medicine Institute that helps us look at reclassifying FDA-approved therapeutics. Okay, so I'll start off just describing our facility. We're a full-service facility, so we're able to do all of the ins and outs of creating new models or rederiving models that have been previously archived, things like IVF. Today, of course, the focus of the talk is going to be our work that we do with CRISPR. We make knockouts occasionally, but the majority of our work now is in knock-ins. And then today, I'll focus on how we are making animal models of human mutant alleles, and the majority of which are SNPs, but occasionally we'll get a partial humanization or some larger change. But for the most part, these projects will be SNPs that I'll show you. So for anyone not familiar with how we make rodent models, this is just a quick workflow of what we do. So we take our CRISPR reagents, all of them ordered through Synthego, and we inject them as ribonucleoproteins directly into the pronucleus of a fertilized embryo. We transfer those into what you can think of as a surrogate mother to carry them to term. And then once the pups are born, we extract genomic DNA from the tails, and then we genotype them. And we have several different assays, but our workhorse that you'll see throughout the presentation is really just PCR and then running them out on a polyacrylamide gel to get really fine resolution and look for any CRISPR-induced changes. And then, of course, we confirm everything through sequencing. Okay, so like I said, at UAB, we are really a hub for neurofibromatosis research, and a lot of our work is in neurofibromatosis type 1. The disease itself won't be the focus of the talk today, but I'd like to think of it more as like a platform for the types of modeling that we're able to do, particularly because it is a rare disease, and that's the focus of a lot of my work. So we do a lot of genetic testing for NF1 at UAB, and many times with a genetic disease like this, we come across variants that we're unsure of the impact that they have. So, variants of uncertain significance. And the question is, how can we prove whether or not this is a causative variant? And so we take a lot of these different variants and we look through them. We look for similarities and differences and try to, we can't model everything. So we try to make really intentional decisions about where we want to model them, look at different domains of the protein and so on. One that was really interesting and stuck out is this mutation that leads to a clinical subset of NF1 called Familial Spinal Nf1, where the patients develop tumors down the spinal cord. It's distinct in presentation from traditional NF1. And at UAB, we do have the ability to make rat models in addition to mouse models. So we set out to replicate this particular patient mutation in the rat, and we got the most striking phenotype. This is a spontaneous breast cancer model in the heterozygous state. So this animal has one copy of the patient allele, this R1276Q mutation. The other allele is initially wild type, although we found that in the tumors, there has been a loss of heterozygosity. And what was exciting for us to see was that this model seems to be recapitulating the pleiotropy that is seen in the human patient. So you can see when we dissect out the spinal columns, if the animals are able to age out with this enormous tumor burden, we do get some animals that have nodules along the spine or pheochromocytomas, which is another phenotype seen in NF1 patients. And then you can see over here just how enormous these tumors can get. Okay, so as we're going through this process of creating animal models of these rare variants, we end up with this entire suite of NF1 rodent models that there for us to use, and we openly and willingly share them with other groups. But what's been really interesting is that now that we've built this suite of animal models, we are not just looking at these models as individuals anymore, but we can look at them more as classes for treatments with therapeutics, and that's exactly what's been done here. So you can see all of the different research initiatives that we have going on at UAB, targeting them more on the basis of the effect that they have on the RNA or protein level. So we have an investigator looking at exon skipping for some of the splice variants, and then or ribozyme editing, nonsense suppression in the case of our stop mutations. And then you can see the different types of approaches that we've had to take. So making these patient variants. As many of you know, you don't have a lot of room for variation in where you can target and the way that you can target. And so we've had to get a little bit creative with some of our approaches. We've had to use alternatives to Cas9 like CPF1 or also known as Cas12a. We've done electroporations. In the case of downstream therapeutic approaches like exon skipping, we really need that entire exon to be humanized because the antisense oligos are going to bind directly to that sequence, and we can't optimize them binding against mouse. So have to make bigger changes in that case. One of the initiatives I'll take a moment to highlight is the research of my long-term mentor, Dr. Robert Kesterson, who's doing nanoparticle gene therapy, and he is using the rat that I just described to you that has that really striking breast cancer phenotype. And so we used CRISPR to make the rat, and now the idea is that we're going to use CRISPR in nanoparticles to go back as a gene therapy and edit that mutant allele back to wild type. So we're fortunate that there's a guide that binds specifically to the mutant allele, and it does not recognize wild type. So once we encapsulate that, we can deliver it directly into the mammary glands and see if we can convert enough of the mutant allele back to wild type that we could have some positive effect on the phenotype. And our first dose with the nanoparticle treatments is actually using mouse wild type cDNA just to test delivery and see what types of levels of expression that we can get. But you can see here, once we encapsulate that wild type mouse cDNA plasmid along with an RFP in the nanoparticles and deliver them, we went in and dissected out the mammary glands, and we're getting really nice expression. So we know we're in the right place. We know we're getting delivery. We know it's expressing. So that's really exciting. Okay, so I'll switch gears just a little bit to focus on CPAM now, although I hope by the end it all ties in together and you can see how nicely these groups flow back and forth. But this is our newly funded center, CPAM for short, Center for Precision Animal Modeling. And this is really, to me, this is the culmination of all the work that we've done at UAB in modeling, not just rare diseases, but any types of patient alleles. So just, we built up this repertoire and this reputation for being able to do it. And then we're able to package that all together, not just in rodents, but with multiple model systems, and become one of 3 centers in the United States that's funded to do this type of work. So there are 5 different units in CPAM, and so you can see how we all go back and forth. Here in the middle is the RSS for short. But what's important to note here is that they're technically 5 distinct units, but we all work extremely collaboratively and are all kind of involved across the board as much as we can be. Okay, so we have 4 different in vivo systems that could potentially be used to model any nominated variant that makes it through the pipeline. So we have worms, fish, mice, and rats. We, in addition to that, also have a cDNA screen that we can do in Xenopus. And we also have an in vitro system. So we can, of course, take any of these patient mutations and put them into a cell culture system and test therapeutics that way as well. Sorry, I'm just having a little trouble advancing. Okay, our timelines here. So, there are differences in how long it takes to make different species and how labor intensive it is and their generation times and so on. So we have our timeline here of when you could expect to see a worm or a fish or a mouse version of the mutation that you're trying to make. And we had to keep this in mind when we were proposing how many different models we could take on per year, what our capacity would be, because it is a lot more expensive and time consuming to make a rat, for example, versus a worm. And I want to take a moment to really emphasize that we are eagerly soliciting external nominations. So this is not a closed system, or this is not a UAB only system. we have a landing page on any browser. There's actually a web form you can access through your cell phone where a physician can nominate a variant for consideration, and really any variant that that is nominated, we will take a look at it and we'll see if it's a good fit for our program. So we really go through them with a fine-tooth comb and make sure that it's something that we can readily take on and model, and something that we can also target therapeutically downstream. In the interest of time, I'm going to breeze through this slide a little bit. I just wanted to show where our nominations come from and emphasize again, we do solicit external nominations as well as the nominations we get through the Precision Medicine Institute or the Undiagnosed Diseases Program and the CCTS network. And so as these come through our workflow, the idea is that we'll be able to get back to the patient at some level and have some type of effect on their clinical care. That's our ultimate goal. So this is our workflow of how we determine if we're going to make a model, and then perhaps, if we're going to make a model, which one is going to be the best. And we really have it's some intense conversations and meetings. So every Monday at 4 we meet and we review these cases, and the fish people will say, I really think this should be a fish, or us rodent people will say, no, it should be a mouse, or we may end up saying that it should be both, and we'll do both of them as long as we have the resources available to do it. And I'll share these slides later so you guys can take a closer look at them. Okay, so hopefully bringing this all together is our NF1 CPAM variant that was approved. Okay, so of course, as we're getting our feet under us with CPAM, we needed to have some demonstration projects. And so we really capitalized on the expertise of existing UAB investigators who maybe had some other types of animal models in that field, or had a rare patient variant in mind that they'd been wanting to work with. And so, of course, one of our overarching themes was RASopathies, which encompasses NF1. And so this same variant that we modeled in the rat, of course, one of the questions I frequently get when I present on this animal model is, does it do the same thing in the mouse? Is this species specific, or is it variant specific? And so to get to the bottom of that, we nominated the variant for CPAM, and as it went through the full workup and workflow as any other variant, it was accepted for us to make a mouse and actually to make a fish of that same variant. So we'll end up with a suite of animal models. And excitingly, we've had some early success in generating these. So this is a mouse. This is the mouse model version of the rat that I showed you earlier. And this is the early CRISPR targeting we're doing for that. So we've got our frequencies down here. We were absolutely delighted to see that of 12 pups born, one of them had the knock-in. And then we went through and sequence confirmed that. So, our systems are up and running. We're getting the sequence confirmed bounders in rodents, and everything seems to be going well. So to give an overview of the number and types of projects that we can take on and what our capacity is, so far 65 projects have been submitted. We're looking for, we have more that we're going to be reviewing, but we're also looking forward to the coming years taking on more and allocating them out, because we have the resources to take on this kind of work. And in addition to creating the animal models, we have an entire unit dedicated to sharing them. So we're going to be depositing them to national databases or sending them out to anyone who wants them. So a lot of work to do. Hopefully, I have shown you how we're having an impact on clinical care through studying rare disease variants at UAB through all of our different units that work together. And I'll take a quick moment to acknowledge everyone in my lab. I don't know where my slides went, but just acknowledge everyone who works in our facility. There they go. And also take a second just to say that we do have a microinjectionist position open. So if you have any expertise in that area, please contact me. I'd love to talk to you. So I don't know if we have time, but I will take your questions if we do. Okay. Can I provide gene editing services internationally. So we certainly can do that through TGEMS. CPAM is an NIH-funded center, so that would be restricted, but we have made plenty of models through TGEMS for international institutes, particularly in India so far, but that's not an issue. I mean, there's a lot of legalese to get through, but we're able to do it, sure. Okay, well then, with that being said, I'm going to close out this session. I look forward to hearing from anyone who wants to talk further, and thank you all for attending all of these talks.